All right, in this chapter, we're going to start really talking about the cell and the structure of bacteria. And this is kind of where I feel like micro really starts. We're going to revisit those three theories from before: spontaneous generation, biogenesis, and the endosymbiotic theory first, starting with spontaneous generation. You're going to read about an experiment done by Reddy, who did a really super simple experiment. Just took 2 jars, put like pork chops in each jar, and then covered one and left the other uncovered. And what he observed was that something was blocking the maggots from basically being born. I would say that this experiment did not prove anything, but it did disprove that life was coming from inanimate matter, which was kind of the falsification of spontaneous generation. Pasteur was really much more important, in my opinion, because his experiment with these swan-necked flasks proved that microorganisms exist and that life comes from already existing life. So this experiment kind of looks like this. There's a funny flask-shaped like glass object that has broth in it. He sterilized it with fire and then he let it cool, and then he tipped some of that liquid into the U shape of the flask, and then he just watched it. And what he saw was that it was still clear and sterile in the base of the flask, but the neck part that was in contact with the air became cloudy and gross and contaminated. And it basically stayed like that for a really long time. And then at some point he decided to end the experiment, tipped that fluid back, and then it kind of came back to life, which is kind of interesting. I will just point out a couple things about Pasteur's experiment. He was pretty lucky that he chose an animal broth, because if he'd chosen a vegetable broth, if it had any soil bacteria in there, they could have formed those stress-resistant endospores that would have survived, and he could have been probably pretty confused. But I will say he was lucky, and he sort of started out the science of microscopic, too small to see with the naked eye organisms that are causing all of this contamination. That brings us to our modern theory of biogenesis, which is our current understanding. And then as part of biogenesis, we look at cells as being the basic unit of life. So let's just kind of review cell theory for a second. That guy Hooke used a magnifying glass to look at plants. He termed this word cell, and that's sort of our unit of organization that we use to talk about a single organism. You should know that it wasn't a super popular theory in the 1600s. It wasn't really for a couple hundred more years before it gained any kind of popularity. And these dudes were the people who kind of like found this old book with this proposal of cell theory, and then it started to take off from there. But it is still very much how we look at the organization of life today. The endosymbiotic theory is something I really want you to think about. It's this theory that mitochondria and chloroplasts are actually microbes that have been sort of cohabiting within another cell. And there's some really cool things about mitochondria and chloroplast that support this theory. This is just a table of the evidence that supports this theory. The first is actual DNA evidence. The DNA inside the mitochondria and the chloroplast is circular. It's not in chromosomes, which is what we see in normal genetic material of like a eukaryotic organism, or like you, for example, have chromosomes. So mitochondria and chloroplasts have their own weird DNA, and it's in the shape that we see in the bacterial world. They don't have histones. That's something that only eukaryotic organisms have. And their protein synthesis, it's very much bacterial in how it works. Like, it always starts with formylmethionine, which is an amino acid that bacteria also start to produce a protein with. Eukaryotic protein synthesis always uses a different amino acid to start. So that's weird. And then they have bacterial ribosomes. Like, ribosomes are categorized based on weight, and our ribosomes are actually a little bit heavier than bacterial ribosomes. They're just a little smaller. But mitochondria and chloroplasts have the same size of ribosomes. And you probably know that mitochondria don't actually undergo mitosis. They just duplicate with a process called binary fission, which is exactly what we see in the prokaryotic cell. So bacteria do that. So when you think about this theory, it's kind of like we live together and the mitochondria and the chloroplast produce all of this energy for our cells and then allow cells to basically specialize because they don't have to make their own energy. I really think this theory is cool and I want you to think about it. And when you need to refer to evidence that supports the theory of endosymbiosis of bacterial mitochondria and chloroplasts, refer to this chart. Oh, and I'm going to add just one thing. Mitochondria have true motility, which is cool. They're totally squirming around inside the cell. So if you thought they were just stuck in the cytoplasm, think again. Now, before we kind of get into the anatomy, I just kind of want to define the word prokaryote and eukaryote. Pro means before, eu means true. And kary is referring to a kernel or like a density inside the cell that they didn't really know what it was when they named it that, but it's what we call the nucleus. So things that don't have a nucleus are prokaryotes and things that do are eukaryotes. When you kind of do a side-by-side comparison, a lot of the differences are no nucleus, no like histones, no chromosomes, none of that. Basically, prokaryotes have very simple anatomy. They just have cytoplasm with a few loose ribosomes. And then eukaryotes are the things that have all of the organelles, like the endoplasmic reticulum and all the things you learn about in a general biology course. They're the only types of organism that has all that stuff. And they undergo mitosis, where prokaryotes just copy and paste, which is binary fission. There isn't even any steps. They literally just duplicate and divide. It's always good to revisit the prokaryotic shapes. Remember, we have a sphere or a coccus, a bacillus, rod, spirals. We'll kind of talk more about spirals in this chapter. There are some star, square, triangle shapes. Those are fairly unusual, and I would say that they're much more likely to be found in like fresh water, like warm, fresh pond water or something. They don't really come up a lot in a clinical sense. So we'll kind of dwell mostly on the basic shapes, the coccus, bacillus, and spiral. But the good thing is that usually bacteria have a designated shape, or they are monomorphic. They have a single shape. A few of them will be pleomorphs, and that just means they can change their shape, and I will always mention what those are. And there's a really helpful table in your textbook to kind of practice with those shapes. Arrangements, always good to touch base on those. You can have a single coccus, you can have pairs, diplococci. Chains, remember, are strepto, like a chain of pearls. Tetrads are 4 cocci that kind of look like they're in a square pattern. They're super cool. And sometimes you can have 2 tetrads that are stuck together like a cube, which is a sarcinae. And very common are going to be clusters of cocci, which are staphylococci. Bacilli come in singles, single bacillus. You can have diplobacilli as a pair. Notice that they're end on end. And then there's streptobacilli, which are like chains, very similar to streptococci. There is no staphylobacilli arrangement. When a rod replicates and it stays attached, it does so like end on end. It doesn't like randomly stay stuck together like cocci do. So if you see something that looks like staphylobacilli, you should just know that it's a pile. It's not actually physically attached to itself that way. These arrangements for rods are like literally just attachments at their very terminal end that keeps them in that arrangement. And then of course, that little coccobacillus is an option too. If you can't decide if it's a sphere or a rod, call it something in the middle. Vibrios have that single curve. Spirals and spirochetes. Here's where spirilla and spirochetes are different. Spirochetes are flexible when they move. They literally kind of bend as they like swim around. And spirilla are perfectly rigid, so they never flex or bend and move that way. So how you would determine the difference, because they both look like curls, is by watching their motility. And if it flexes, it would be a spirochete. Here's the star-shaped bacteria, which is kind of rare. It's sort of a weird lumpy star. And then we've got sort of that rectangular halo arcula, which kind of was mistaken for a plant cell for a very long time, but they're fairly rectangular, I guess. And now we can actually look at some of the actual anatomy. So we're going to work our way from the outside of the bacteria in, starting with a potential kind of sticky outer surface, and then we'll work our way through the cell wall, plasma membrane, into the cytoplasm. So let's start with the outside sticky layer that might be there. Glyco is a term that means carbohydrate or sugar, and calyx means shell. So it's a shell of carbohydrates, and the way that I think about this is like sticky sugar. It's wet sugar like a lollipop, and it tells you what the function of that structure is. It's actually going to make this a really sticky outer surface, and the glycocalyx is going to have two potential forms. It can be tight or it can be loose. If it's tight, we would refer to that as a capsule. If it's loose, we would refer to that as a slime layer. So here's a picture of those two options. The image on the left is a capsule stain, and the image on the right is a scanning electron micrograph. So on the left, you can see that there's this clear halo around those little streptococci, diplococci, and singlecocci. That indicates that the carbohydrate sphere is really composed and tight, but it is very sticky. On the right, you can see that there's something really kind of mucousy and slimy about that SEM, and it's similar in that it's sticky and it's full of sugar, but it's just really loose. So the function of the capsule in the slime is the same. It still allows these microbes to be sticky. It's just one is nice and tight and one is really loose. Bacteria can have a flagella. It works exactly the same as it would in any other cell. It's for spinning and basically like propulsion. But what's interesting about bacterial flagella is they can have more than one. So we need to refer to their arrangement or their number and position of flagella rather than just do they have them or not. So let's actually look at a picture and then I'll go back to those terms. The first image on the left is a monotrichus. Trichus means tail. So mono, single tail. This is a really beautiful bacillus with a single flagella, monotrichus flagellar arrangement on one end. Next we have amphitrichus. Amphi means both, and it has a single flagella on both ends. LOFO is a bundle. So there's a bundle of flagella, and in this image, it's on one end. But the trick about Lophotrichus is that it can have a bundle of flagella on one end or on both ends. So keeping in mind that that one can kind of be illustrated either way. Peri means around, and this one on the right is kind of probably moving a little bit, so they're not like kind of radiating all around, but it would be completely covered in flagella if it was peritrichus. I read this paper not too long ago, and it was actually really cool because I've always read about flagella in terms of swimming, and that's sort of the typical way of thinking about them. But in this particular paper, they were talking about an organism known as Pseudomonas aeruginosa. We will talk about that later. It's one of the common complications from people who've had burns over large parts of their body is an infection from this bacteria. It's really nasty. But it stands up and walks on its flagella, which is so cool because one of the things about this bacteria is it's really hard to treat. And I think part of the reason why is it just literally gets up and walks around. So, with flagella, we produce motility, and motility is movement with purpose. But I want you to know that bacteria don't really have like sensors or a brain or anything, so they don't necessarily know where they're going. They kind of have two types of movements. They're called tumbles and runs. A tumble, it kind of like sounds like it's tumbling or it's sort of spinning. That's kind of where it's reassessing its environment, and then it can only move in a straight line, which is a run. So it can go towards things and away from things, We know that it can kind of sense at least different types of stimuli that might be chemicals or light, gravity. It's weird because I don't really have like eyes or anything. But we do kind of have to describe their true motility in this way. Tumble, they're just kind of spinning. Running, they're moving in a straight line. And on the bottom of this slide are two different scenarios. The one on the right with the red is a gradient of some kind of concentration of attraction, like maybe food. And what you'll see is they make really long runs and just a couple of tumbles. And they might not be going in a perfect direction, but overall they're going to be getting towards that attraction area. The one on the left is also really important, and I want you to look at it for a second. I want you to notice how short the runs are, because in this situation there's no gradient, there's nothing there. It's just random environment, and when that happens, they just make lots of tumbles. So they don't really move very far. They keep looking for something interesting, and they should never be moving in a direction. They kind of move really randomly, and that's how you tell the difference of whether or not it's attracted or repulsed from something, is the length of those runs and the frequency of the tumbles. Now, an axial filament is actually just a flagella, so don't get confused. It sounds different, but it's a flagella. But the flagella, instead of being on the outside, is actually on the inside of the cell. And as the flagella turns, it coils up the bacteria and it causes them to actually spin and rotate that way. So this is what makes spiral bacteria, and as they have motility, they actually just kind of like barrel roll through the environment. But it's kind of cool that it's actually the flagella that like turns and creates that spiral morphology of spirilla and spirochetes. Next are two structures, fimbriae and pili. So take a look at this electron micrograph for a second. You can see a really nice rod, a nice bacillus, and there's definitely a dark kind of corded flagella. That's not what we're looking at. We're kind of looking at that kind of hollow tube, the kind of thicker but not quite solid tube. That's the pilus, and there's just one in this example. And then those kind of weird hairs that are sticking out of the bacillus around it, those are the fimbriae. So fimbriae are like little teeny tiny finger-like stickers. So if there's no capsule or slime or anything that makes it stick, they could have fimbriae, and they sort of have like these little hair-like stickers that they can use to colonize your body's mucous membranes, which are like your body openings, basically, like your nose or your mouth. So that is a good like quality for pathogens. And then the pilus or pili is a hollow tube for sharing DNA. So these microbes that have this structure can basically hook up with one another, and they can just share genetic material that way. And it doesn't have to be species specific. So fimbriae and pili are both clinically significant because they can increase the potential pathogenicity or the ability to evolve and like genetically share like toxins or antibiotic resistance, for example. And that doesn't even have to be between related species. They can just like hook up randomly. Next is the cell wall, and this is kind of a big important point for us. The cell wall is really special in bacteria because it is made of peptidoglycan, which is a super unique substance that we only see in bacteria. And because it's unique, it's a target for a lot of our antibiotics. So penicillin, for example, is a classic antibiotic that targets peptidoglycan, and it's very effective. But it does provide a little bit of shape and structure for the actual bacteria, but we just want to look at it on a more molecular basis. So peptidoglycan is these really square, geometrically arranged molecules of alternating polymers. It's N-acetylglucosamine and N-acetylmuramic acid. Just know that there's two molecules that are every other one, and then there's these little proteins or polypeptides that kind of hold it into a shape. Now, this is a picture from your book, and what I want you to be visualizing here is that gram-positive bacteria, the ones that stain purple, have a thick cell wall, which means they have layer after layer after layer after layer of peptidoglycan. There's also an additional molecule called teichoic acid that kind of helps stabilize that thick cell wall. Gram-negative bacteria have a really thin single layer of peptidoglycan. They do not have teichoic acids. And then they have a weird outer membrane on the surface. So I'm going to show you a different picture that's not in your book. And I think this is helpful to visualize first. The example on the top is a thick multi-layered structure of peptidoglycan and then teichoic acid. So that's what the gram-positive cell wall looks like. And on the bottom, you can see there's kind of rods of peptidoglycan, but then there's an outer lipid membrane outside of that, which is so weird because it has a double membrane. And that outer membrane actually has some important structures that we need to talk about. So first, let's kind of look at a gram-positive composition. We have lots of peptidoglycan, and it's either going to be attached to the cell wall, which is the wall is sort of the lipid bilayer membrane. And then we have like layer, 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 layer with teichoic acids kind of stabilizing it to itself and to the plasma membrane. So that is unique because it's thick peptidoglycan with teichoic acids, and it's going to give it a really like thick outer structure for protection. The term antigenic variation means that it's a surface molecule of this cell, and an antigen is always like an outside structure of any cell, and this can kind of vary a little bit, which makes these bacteria sneaky for your immune system to try to recognize, which is why you can't always get rid of gram-positive bacterial infections by yourself. Now, the gram-negative one has more complexity, in my opinion. It's got a normal plasma membrane, a single layer of peptidoglycan, and then it has an outer membrane, which we call the outer LPS. That outer LPS has a couple things I want to mention. One is a antigen, so that's kind of its surface identification for itself. And then it has actually an endotoxin as part of that outer membrane. And that outer toxin is really important in microbiology because as that bacterial cell dies, it releases that endotoxin, and that can be a clinical event of toxemia, which can be horrible. So gram-positive bacteria don't have that. I mean, they might produce a different type of toxin, but it's not a toxin that's released when the cell dies. Usually, it's a metabolic toxin that if you kill the bacteria, it's unable to produce any more toxin. Whereas with gram-negative bacteria, you kill it, you release this endotoxin, which is really weird. So now I want to think about the gram stain for a second. So the gram stain is where you have a first crystal violet stain staining all of the cells. And then you add the mordant, that special enhancer iodine, which links to the crystal violet and forms like a really large molecular crystal. And then you use alcohol and decolorize those cells, which means it dissolves the outer second membrane of gram-negative bacteria, and then that whole crystal violet iodine complex just leaks right out, and then it's invisible again. And then it's still trapped inside the gram-positive cell. And then you add a counterstain so that you can see the red gram-negative cells. But like, you should now see how this gram stain can tell you something about the anatomy of these cells. In terms of damage to cell walls, it's a classic drug action mechanism to go after peptidoglycan, and it's a good one because people don't have peptidoglycan, so there's no potential side effects. Penicillin is the #1 classic example of an antibiotic that basically goes after peptidoglycan. And I think you could probably see at this point why penicillin is appropriate to treat gram-positive bacterial infections, but not necessarily gram-negative because they don't have that much peptidoglycan. Your body also produces a substance called lysozyme. It's in your saliva, it's in your body juices, it's in your digestive juices, and it also acts like penicillin, which is great because when you put food in your mouth, there's gram-positive bacteria in sort of the skin and all over the surfaces, but you don't really want gram-positive bacteria in your gut, so you have all these like chemicals like lysozyme that basically kills those surface bacteria and allows the gram-negative gut bacteria to grow. In terms of the plasma membrane, the lipid bilayer, there's really nothing special about bacterial plasma membranes. They kind of have all the same properties as every other type of plasma membrane you've ever studied, but it's a, semi-permeable like structure. It's going to control things through channels. Literally nothing special about it. It's just they have plasma membranes the same way we do. Here's a picture. It totally looks exactly like, I could probably use this for any example of any plasma membrane. Inclusions can be anything from organic to inorganic, and I actually think inclusions are really interesting. I think it's interesting that they're attracted to things like magnets. It makes me wonder if someday you could put magnet therapy into an infection treatment protocol and maybe like magnetically suck bacteria out of a wound or something. But I will tell you, these are not well understood, but it is interesting that microbes collect things. Here are some weird inclusions, like you'll just kind of see little dots and specks inside the cells, and they just look like weird things, and we can't really say too much about them other than bacteria like to collect things the way we do. Endospores are kind of neat. Like if you have an endosporulating organism, they basically are going to stress out, and then in a matter of minutes, you guys, it takes like 5 minutes for them to just pack all of their important pieces into this protective little cystic cortex, and then everything outside of that will just disintegrate. And then the endospores will survive for a really long time. Like as long as the stress is there, they are gonna basically be protected, and then they hatch out. So it's true that we have some terminology to describe endospores. Like if they're just hanging out, they're like at rest. But when they are trying to protect themselves from desiccation, heat, chemicals, antibiotics, for example, they will sporulate. And then after the stress is gone, they return, which is germination. There's one more thing that I'm going to add to this chapter, and it's not in your textbook, but I think it's really important, and it's that they have little teeny tiny microscopic wires that actually attach to one another, and it allows them to electrically communicate. And this will be important because one of the things that's really cool about bacteria is they can coordinate release of toxins so that they all do it at exactly the same time. And it involves something electrical, and we didn't notice these structures for a very long time because they're so tiny. They're like nanometers. So we needed high power microscopy basically to even see them. But we will see that they're capable of communicating and doing some pretty cool like behavior that way.